afterwards. Anyway, welcome everybody to the Grantham Institute. I recognise most of the faces here, but for those I don't, you're very welcome here. Grantham Institute is one of the college's grand challenge institutes, and we try and encourage and stimulate research and all sorts of activities around climate change and environment across Imperial College. You may have seen our banners. We are now 10 years old, and we've got a range of activities going on this year to celebrate that. We'd be very grateful if there's a register, if you'd like to sign the register so we can just see a record of people who come to our events. That's great. And anybody who's not signed up for our mailing list, please go on our website and do that. Anyway, that's enough about us. We're very pleased today to have Steve Long here. He's a really eminent in his field. He's the Gutzgeller Endowed Professor in the Department of Plant Biology and Crop Sciences at the University of Illinois, and Professor of Crop Sciences at Lancaster, and visiting Professor in Lincoln College and Plant Sciences in Oxford. Yeah. So he's three people in once. His research bioengineers the photosynthesis process in crops to achieve higher productivity, sustainability, and adaptation to climate change. He heads an international project to improve the crops that feed many of the poorest in the world, and has led to the discovery of a way to engineer photosynthesis that resulted in a 20% increase in crop productivity. Steve is a Fellow of the Royal Society and both of the American Academy for the Advancement of Science and the American Society for Plant Biology. His CV is much longer than that, but that's it in a nutshell. So thanks very much for being here, Steve.
Thanks. Thank you very much for the invitation. I think it's 30 years since I last spoke at Imperial, so thank you for inviting me back. This is my home for most of the year, the Institute of Genomic Biology at the University of Illinois. But I just point out right in front of it is part of our experimental farm. We have 6 square miles of experimental farm. This time of year in Illinois, about 60% of the state is covered with either wheat, maize or soybean. Illinois accounts for about 10% of global production of corn and about 10% of global production of soybeans. So we're very sort of tied to agriculture around us. And although in here we've got all the modern tools of plant molecular biology, a lot of it is related to translating it into field crops. Another very important facility on the campus for me is the National Centre for Supercomputer Applications. This might look like a building, but it's actually supercomputer, Blue Waters, which is full of cores. It's very important to the in silico side of the work we've been doing. And as was mentioned, I have a part-time position at Lancaster, where we're really interested in how we can not only engineer productivity increases, but also achieve those increases without using more water. And Lancaster has a lot of facilities for high throughput phenotyping of water use in crops. And as was mentioned, this year I've been given sanctuary from our political situation in the United States at Oxford, but sadly that's coming to an end. So what I want to talk to you about briefly is rising world food insecurity, why I think photosynthesis is a solution to this, or an important part of future solutions, mathematical modelling to leading to that, and then proof of concept, crops engineered to be more efficient. And I want to make a few final words about bioenergy as well. So rising world food insecurity. About 2/3 of calories in human diets globally come from just 4 crops, directly or indirectly. And that is maize, rice, wheat, and soy. Obviously, rice and wheat, we might argue, are the most important because they are direct sources of human calories. A lot of the maize is going into animal feed, although it is a very important direct food source, particularly in Africa. Soy is the largest vegetable protein source globally. Now, if we look at, we've been in a situation where global yields have been improving, nutrition has been improving, but that has reversed in the last couple of years. And in the latest FAO report, they're pointing out that the number of people who are food insecure has increased over the last 2 years by quite significant numbers. And they also point out that the trend is towards this worsening. And why might that be? Well, if we look at what we've achieved from 1960 to today, this is rice and wheat. There's really been globally, this is yield per hectare of land. Globally, that's steadily increased, or if you plot it out, it appears to show a linear increase. So if we project forward and we say, well, if we can continue to do this, then by 2050, whoops, sorry about that. 2050, we would end up round about here with nearly 5 tons per hectare of wheat, just over 6 tons per hectare of rice. So that would assume we're getting increased agronomy, improved genetics. But actually what FAO say we need this is the trajectory they say we need. Their prediction is that we will need about 60% more of these primary foodstuffs by 2050. That's of course because global population is rising, but actually a bigger driver is that the global population is becoming more urban. Urban diets tend to demand more calories because now you've got to transport food, you've got to store food, you've got to retail food and so on. All results in more wastage. Because urban populations do tend to be more wealthy, there's also an increasing demand for dairy, for meat. So diets becoming more Western style, demanding more primary foodstuffs. And of course, there is volatility though. You know, what this is projecting is a growing gap between demand and supply. But of course there's stochasticity in this, and this is from the FAO report where they're pointing out, well, 2008 and 2011, actually there were, if we look at wheat, there were some poor harvests in some of the major producing countries. That resulted in other countries shutting down their exports, which led to quite strong elevations of wheat prices. And FAO have tied this in with food riots. Each of these red lines is a food riot somewhere. And of course now those food riots have been tied in to the Arab Spring, which is why in the United States, the Department of Defense is more concerned about food security than the Department of Agriculture. So now of course, if we look forward to where we might get by 2050, we might meet this demand by reducing demand, eating less, increasing production, and avoiding those losses. Maybe diets become more vegetarian. But nevertheless, we should take account of the fact that actually all of these trends are in the opposite direction globally. So although theoretically we could achieve this with fewer losses, more vegetarian diets, we shouldn't bank on that. We should be taking out insurance policies against it. And then there is the elephant in the room, climate change. We do know now from experiments, particularly with wheat, that just a 2-degree rise in temperature in the Australian wheat belt would result in a 50% depression in production. We can see that from year-to-year climate variation. And this is not taken into account in those projections. Now, of course, we could get more food by taking yet more and destroying yet more natural vegetation, A general trend is when prices rise globally, there is incentivization, legal or illegal, to destroy more forest, plant more land. So what we need to achieve is more yield on the land we're already using. Now, I should say that I showed those nice linear rises, but actually if we dig deeper into that, really it's not quite the case. China, India and Indonesia are the biggest rice producers in the world. In the 70s and 80s, these countries were increasing their yield per hectare of land by about 20 to 30% per decade. If we come to the first decade of this century, then the increase is less than 10% per decade. And what is striking about this is that China was investing 100 times more in the first decade of this century to what it was invested in the 70s and 80s in improving rice productivity. So this is telling us it's getting much harder to get yield increases than it was in these years, which were the years of the Green Revolution. So why might that be, and why might photosynthesis be one factor we can draw in to try and get some yield increases? Well, if we look at genetic yield potential, this is what a cultivar of a crop can achieve on a piece of land if it's without disease and pests, if it has the nutrition and water that it actually needs. So that's determined by the amount of visible solar radiation, because photosynthesis uses visible radiation pretty well only, that's available over the growing season, multiplied by how efficient the crop is at capturing that solar radiation, multiplied by how efficient it is at converting the captured radiation into biomass energy, how good it then is at partitioning that biomass into the part of the crop we care about, so the grain of rice, for example. Now, these are figures for a modern soybean cultivar growing on our university farm, where we have good soils, good rainfall. We don't fertilize with nitrogen because it's a leguminous crop. But basically, if you look at this, a visible radiation over the growing season, it's intercepting almost 90% of that radiation. If we look at partitioning efficiency, how much of the biomass ends up in the seed, that's 60%. So these numbers are already remarkably high. The conversion efficiency, though, which is primarily photosynthesis, is just 3%. So how do these numbers compare to theoretical? And what are our prospects for trying to improve on those? Well, what I want to do first of all is take you back to what a soybean or a rice plant would have done before the Green Revolution. So pre-Green Revolution cultivars would achieve about 60% radiation interception efficiency, i.e. of what we think is the theoretical maximum. Their partitioning efficiency was around about 30 to 40% of the theoretical. Photosynthesis was around about 10% of the theoretical efficiency. If we take the plant process, look at each step, the energy, theoretical energy losses, then what the crop is achieving is about 10% of what we think the theoretical could be. Now, if we look at what the Green Revolution did, it really closed the gaps on interception efficiency and partitioning efficiency. So said today, partitioning efficiency in our modern cultivars of soybean, wheat and rice is round about 60%. So if we're going to have some stem, some husk to hold the seed, we're not going to get much higher than that. But if we look at improvement in conversion efficiency over the Green Revolution, it was almost unchanged. So really, conversion efficiency is the one thing we have left that we could change. This, of course, is the genetics. That doesn't mean that we can't get yield improvements through better agronomy, disease and pest resistance. Well, if we were to improve photosynthesis, do we know we get a yield increase? Well, these are experiments that I was able to set up when I first moved to Illinois, where we're looking at the impact of elevated carbon dioxide on the crop. And we wanted to do this under open air conditions because you can't really do it in a greenhouse, a closed environment. No field crop in a greenhouse looks the same, behaves the same as it does on an open air condition. So this is pretty well in the center of the corn soybean belt in Illinois. And what we've done here on an experimental farm is this ring. Within this ring, the carbon dioxide level is elevated to the levels we think we're going to get between 2050 and 2060. So we do that. If the wind is blowing in this direction, carbon dioxide is released from these pipes according to wind speed and direction. So the higher the wind speed, the more CO2 we have to release. That's carried across the plot, so we get an elevation of CO2 within this plot. And here we're measuring wind speed and direction and also CO2 levels to make sure the computer algorithm is actually getting it correct. So if it puts in too much CO2, the CO2 sensor backs off the valves. Too little, it opens them up. These rings do release about 4 tons of CO2 a day on average to get that elevation. But we're not causing global change. We're taking CO2 from an ethanol plant in Decatur, and instead of it being released in Decatur, it's being released on our site. So we're not the cause of rising CO2. But of course, CO2 is a limiting substrate for photosynthesis in C3 crops. So that includes soybean, rice, and wheat. So the major effect you get of releasing CO2 under these well-watered conditions, because we've got high rainfall and deep soils, is to boost photosynthesis in this crop by about 25%. I should say we elevate CO2 from sowing right through to dry down of the crop. I made it sound quite simple, but it actually took the engineers a whole year to really perfect this system, and it's been running since 2001 now. But what does that do to yield? Well, in soybean, it boosts yield by 16%, and similarly in rice and wheat. So we know, even with a current cultivar, if we could boost photosynthesis in some way, we will get more yield. So I said in the Green Revolution we haven't improved photosynthesis. So why do we think we could do that now? Well, first of all, photosynthesis is probably the best known of all plant processes. We know pretty well every step of the process. We know the genes underlying all of the proteins involved in this. We know quite a lot about the kinetics of the enzymes involved. So we've got a great deal of detail. We can simulate much of the process that's going on in the crop leaf with high performance computing. We can then probe that with optimization routines to say, well, are resources optimally invested to get the most, if you like, CO2 fixation per unit of investment. And then crop transformation, i.e. altering, putting in more of an enzyme, less of an enzyme, putting in alternative pathways is becoming more and more easy and becoming more routine. So we, particularly in the early 2000s, using the supercomputing facilities at Illinois, we did quite a lot of this mathematical modeling. And it's basically like looking at a car production line. You've got 1,000 workers. Where do you put them in that line to get the maximum output? And of course you can do it experimentally by moving people around. But if you simulate it on even with 1,000 workers, you end up with billions of possible permutations. So a supercomputer can work through those very rapidly and give you a solution. So we set about doing, this is the discrete steps in the photosynthetic process. We set about representing each of these as a differential equation to get a system of differential equations for which there obviously wasn't an analytical solution. We then used numerical integration to get that system going. And you might wonder, why on earth is a rocket here? It results in a classical stiff system, which is very difficult to numerically integrate. And when we were talking about this at the supercomputing center, one of the mathematicians said, oh, I have exactly the same problem in balancing rocket motors. And so he helped us with his algorithms, and it actually started to work. But he did say, actually, our problem was harder than his. So just like to tell you that plant biology isn't rocket science. It's harder than rocket science. And that's why we have a rocket scientist as our middle author on the first paper. But what we then used the system to do was to apply an evolutionary algorithm to say, well, okay, the plant has invested its protein this amount in each of these steps. If you've got that amount of protein, is that the best way to invest it? Now, there's an additional problem here in that leaves are not in the same environment all the time. Over the course of a day, the light is changing all of the time. And during the life of a plant, a leaf which starts out in full sunlight here, as the crop canopy grows, is then in shade. So optimizing the system has to take account of this as well. So we set about looking at a number of things that we thought could be changed from the literature and modeling those, and then coming up with estimates of efficiency gains. And this was a paper based on that. But at about this time, the Gates Foundation started to read these papers and then called us up and said, well, do you think any of this could be done? And we said, well, we're not sure, but we'd like to try. So, of course, models like this can look very attractive, but when you've got to try and reduce it down to growing plants in the field, it can look a lot worse. But nevertheless, we decided we'd step up to the challenge the Gates Foundation had made and managed to get a team together that was willing to go where no sensible plant biologist has gone before. And this was really our leadership team. This is Martin Parry, who was at Rothamsted but is now at Lancaster, Donald at USDA, Martin Parry at the Australian National University, and Christine Rains at the University of Essex. So these are my co-conspirators. And so basically after a year's discussion with the Gates Foundation, they decided, well, we'll put our money where your mouth is and you show us this can be done. And so this was really our philosophy, that we use the modeling to predict where we might intervene. And then we do that in tobacco. And you might say, why on earth tobacco? It's hardly a food crop the Gates Foundation should care about. The reason why it's tobacco is very easy to genetically transform. And it produces a lot of seed, unlike rice, soybean, and wheat. So basically we can go from a genetic transformation through greenhouse trials to a replicated field trial in 12 months. And tobacco, of course, produces a full canopy like wheat or rice or soybean, which is very important because you're not looking just at these leaves which are in full sunlight, but these leaves which are in shade are also contributing a large amount of the carbon to the crop. So then tobacco was our filter. If the replicated field trial shows that this technology really improves things, then we would go on to crops the Gates Foundation care about, which is cowpea, which is a very important vegetable protein source in sub-Saharan Africa, rice, also soybean, which is becoming more and more important in sub-Saharan Africa, and maize as well. So we had 7 different pipelines, different ways in which we thought we could improve photosynthesis. And we now have field trial results which show that 3 of these are working. And I'll just tell you about a couple of those. So first one is, one of the early results of our modelling was we looked at the enzymes of the Calvin cycle. This is the cycle which, after CO2 is assimilated, then takes that carbon dioxide and produces sugar and regenerates the CO2 acceptor. And in black is the amount of protein that was present in the leaf. But the computer optimization said, well, if you've got this amount of protein, this is where it should be. And this one really stuck out, this enzyme, C-deheptulose bisphosphatase. There's not much of this enzyme present. This is a logarithmic scale. And the computer was saying, well, there should be 7 to 8 times more than there is. So our colleague, Christine Rains, had already tested this in tobacco. So here she's put more of genetically engineered these tobacco plants separately to express more of this enzyme. And in the greenhouse, you can see it's producing. These are much more productive plants. So we went back to our computer simulation to wonder, well, why could that be? Why hasn't this already happened? And so one of the things that was suggested to us was, well, these plants evolved in a CO2 concentration of about 200 parts per million. That's the average of the last 25 million years. But today, the average concentration of the globe is over 400. And half of that CO2 increase has occurred in just the last 50 years, which in evolutionary times, evolutionary scale, is nowhere near enough time for evolution to catch up. And indeed, when we run this optimization routine at that CO2 concentration, it said this enzyme had no control. Increasing it would have little effect. But as CO2 goes up, it has more and more control. So we had a chance to test that. We then put that into our elevated CO2 facility in Illinois. And indeed, Christine's plants in the field grew better. But when we increased CO2, her plants did even better. The modelling also predicted that temperature rise would give a benefit by elevating this enzyme. And so my colleague, Carl Benaki, also put in open air temperature increase. And indeed, we did find an interaction between CO2 temperature with this engineered increase. Now, I don't want to give you the impression that CO2 and temperature are all going to make crop yields better. What it actually did was protect the plant against the yield loss, which resulted from a 2-degree temperature rise. So in fact, we weren't getting more, we were just getting what we were getting today, but avoiding the loss in yield that resulted from this temperature increase. And although CO2 results in a yield increase, it also has some detrimental effects on the crop. So I'm not saying CO2 and temperature rise are going to help food production, they're not. But we did find a way in which we might be able to protect, at least do some proofing against these changes. Now, of course, when you remove one bottleneck, another one appears. And so in the modelling, then said, well, this enzyme's probably the next target. And indeed, Christine has now done this. So here is one elevation, here is the second one. And again, in the greenhouse, she was getting more productive plants. And this summer we were able to, last summer, we trialed those in Illinois, and that also gave an increase. So I want to tell you about a second pipeline which has given us quite a large increase. Most crop leaves, when they're in full sunlight, are receiving more sunlight than they can use. This is primarily because carbon metabolism there's just not enough capacity there to use all of the light energy which is coming in. And there are some involved reasons for that. But basically, this energy is being absorbed by the chlorophyll molecules. It puts the chlorophyll molecules into an excited state. And if they can't dissipate that energy in some way, those excited state chlorophyll molecules could react with oxygen to produce singlet oxygen, and then a range of oxidizing radicals, which would literally bleach the leaf. So of course leaves protect themselves against this, and they do this by inducing a process which is not fully understood, but results in that excess energy being harmlessly dissipated as heat, a small amount dissipated as fluorescence. Now when that leaf goes into the shade, now it could use all of that light energy in photosynthesis. But initially, when it's shaded, it still carries on dissipating quite a large chunk of that energy as heat. And it takes many minutes before this process relaxes, so that it's now using most of that energy in photosynthesis. So during this change, there's a lot of loss, if you like, of efficiency. So basically, this is rate of CO2 uptake in photosynthesis in the light. Of course, when it goes into the shade, it drops because there's less light. But actually, it drops far lower than it needs to. And it takes many minutes to recover back to a steady state. So this is all lost potential productivity. And you might say, well, this is important in an environment where the sun's going in and out of cloud. But actually, it's important in an environment where there's no cloud, because this happens at the level of a chloroplast, a point on the leaf. And over the course of a day, as the sun moves across the sky, these leaves in the canopy are moving in and out of sunlight all day long. So this is the upper layer. Of course, it just follows the solar energy. But the next layer down is going, a point on the leaf is going in and out of shade all day long as the solar angle changes. And if you add all of these up in a crop canopy and you say, well, how much potential carbon uptake am I losing from this slow recovery? It turns out to be, it varies with crop species, but it can be of the order of 20%, in some cases even as much as 40% of potential carbon uptake. So we looked at, from the modeling, discussions with Chris Niogi at the University of California, Berkeley, who we collaborated with for a number of years. We looked at what we might need to change to get a faster recovery. And the modeling suggested we should increase these two enzymes which convert violazanthin to zeaxanthin and back to violazanthin. So we wanted to increase the rate of this cycling. We also knew that this protein altered the amplitude of this heat dissipation as well. So we reasoned we should upregulate these three enzymes from the modeling and what we knew from theory as well. Now, I also mentioned that chlorophyll molecules fluoresce if they're in an excited state. So we could also see if leaves were relaxing faster from fluorescence. So what we could do with our tobacco seedlings, where we had many transformants, where we could pick out the ones which were relaxing most rapidly when we put them into the shade and grow those on. We checked that this is the gene expression. So what we did was to put Arabidopsis genes into tobacco And the red, this is the logarithmic scale. The red is the expression of the Arabidopsis, the grey is the native gene. And we could see that the proteins they were coding for were all upregulated. Most importantly, this is efficiency of CO2 assimilation. And what we can see is that in continuous light, our transform plants here in the red were not affected, but in fluctuating light, they were twice as efficient. So basically what we'd done was to now speed up this recovery and improve on this loss of efficiency. Now in the greenhouse, these are 3 separate transformers. The plants look larger. So that was great. We now got a lot of seed. We set up a large field trial. And as any farmer will tell you, no year is the same as the previous one. We had the highest rainfall in June ever recorded in central Illinois. We thought we were putting our trials in on flat land, but we were wrong. This end of the field became anoxic, so these plants basically died, completely wrecked the experiment. So that put us back a whole year. Fortunately, the next year and the following year, we managed to get in trials, and what we were able to show was that the productivity of these tobacco plants was increased 2 of the events by over 20%, third event by 14%. So this is huge in terms of agricultural productivity. If a plant breeder gets a 1% improvement, they're pretty happy. So, and this appeared to be the first time this had actually been done with replicated field trials. So of course now we're moving on to putting this into cowpea, as you can see here, into cassava and into rice. We've now got the cowpea where we've got to a state where we can show, at least from the fluorescence, is behaving as it should do. We're now setting up field trials to now see if we can get the same increase in a food crop that matters. Now, of course, one of the questions is, well, we've got several different ways in which we're trying to improve productivity. I mentioned to you about the RUBP regeneration, the SBPAs. I've told you about the work we've done with faster relaxation and non-photochemical quenching. My colleague Don Ort has now also shown a significant increase with improving on the energy used in another process. So we've got these 3 separate events, and we've now been combining these in a crop model to say, well, what would we get if we tried to stack these traits? And that suggests that we could get as much as a 50% yield increase, and we're now looking at ways in which we can try and do that. But that would really be very substantial. All of these processes are not costing more significantly more in terms of nitrogen. In the case of these 2, there's no water penalty either. So we think at least we have the potential to get quite a large yield increase without much more resource. Now, of course, this is all transgenic, so you've got to be able to accept genetically engineered crops to do this. Should say we've also been now looking at the other side of the coin. That is, when a leaf has been in the shade and it comes back into sunlight, it takes time for it to recover. And this is the flag leaf of wheat, shaded by the ear, then coming back into full sunlight. See, it takes several minutes to recover. If we could speed up this recovery, then we've calculated again, taking light over the course of a day, that could probably give you 10 to 15% more carbon fixation. And we've now been able to attribute a lot of this to activation of the carboxylation enzyme, Rubisco. So the group at Lancaster is looking at ways it could try and accelerate that. one of the other things we discovered, and it was a bit unexpected, was with that 3 gene change, of course we've looked at what the single genes have done. One of those genes we looked at was this protein PSBS. And so we've downregulated it to almost nothing with RNAi, and we've upregulated it with extra copies. And what we found to our surprise was there was a very strong correlation between stomatal conductance. So this is the opening of the pores in the leaf, which allow CO2 in and water vapor out, with the change in redox state, which was resulting from us changing the amount of this protein. So that gave us the chance to then actually lower stomatal conductance. As CO2 has risen, we can sort of say, well, actually, we could afford to back off stomatal conductance without much cost to photosynthesis. And so this gave us the opportunity to try and engineer this. And indeed, what we did was, here we've upregulated PSBS relative to the other proteins. And indeed, this is a measure of water use efficiency. We've got a higher water use efficiency without in our field trials a yield loss. So you might say, well, this is all very far from plant breeders in Africa, and I'd say you're right. I don't think these technologies will be available for farmers in Africa at scale for possibly 20 years, but there is some hope. The Australian government has been working closely with the Nigerian government on regulatory framework to get genetically engineered crops into the field in Africa, and indeed it looks quite possible that there be BT cowpea released within the next 12 months, and we've tied in our first trait, this MPQ1, with this pipeline that the Australian and Nigerian government have set up. Finally, I just want to say a few closing words about bioenergy. So I think if we were to accept these changes, we do have the prospect of being able to possibly meet future food demand without bringing yet more land into arable cultivation. Now, of course, it could be that we do get the message, our diets become more vegetarian globally, wastage goes away, we don't need these increases in crop yield, which will be great. But of course, what that would mean is we would then actually need less land for agriculture, which wouldn't be a bad thing. Now, many of the things I've told you would also apply to potential bioenergy crops. And this was a review that we wrote a few years ago now in Science, led by Chris Somerville, on lignocellulosic biofuels. And we think there is a case for these in the United States. If you look at the United States, has almost 1,000 million hectares of land in the 48 contiguous states, so not counting Hawaii and Alaska. About 176 of that is in arable production. About 273 is semi-arid desert. At the time of the Civil War, this number was probably about double that level. We have about 11 million hectares in what we call conservation reserve program, land which is, we pay farmers not to grow food on that land. With a crop like Miscanthus, which is growing it under rain-fed conditions, very low nutrient inputs, you would need about 9.7 million hectares to reach the renewable fuel mandate that was set. So it's a very small number compared to this. With a crop like agave, which could be grown on this semi-arid land, you'd need about 39 million hectares. So I would say these figures are not so ridiculous for a country like the United States. And much of this semi-arid land is actually quite degraded land. A lot of it was land which was overgrazed during early settlement, resulted in desertification of quite a lot of what we call the desert southwest. Crops like this, and there have now been trials in the US, would actually start to put a bit of organic matter back into the soil, would give some protection against land erosion. So this is where brown is where we're growing our arable crops today. But much of this land historically was used for food crops, is now in, a lot of that land is sort of abandoned or in very low intense use. Much of this that could be used for bioenergy production. These are areas where you could certainly grow crops like agave. One of the things we've been looking at is particularly biodiesel, but even more so jet fuel, which is something that is going to be very hard. Aviation fuel is going to be very hard to replace. Now, if we look at our sources of biodiesel today, they're soybean, canola, and oil palm. What we've been looking at in a DOE-funded project is the possibility that sugarcane could be engineered that instead of its stem producing sugar, if you could engineer it, that it put that energy into oil instead of sugar, then this is the amount of barrels you could get per acre compared, for example, to soybean. That makes it, if you could do this, that makes it economically very viable. And indeed, this is work not done by my university, but by Brookhaven National Lab and the University of Florida, where they've actually been able to now put in genes that have resulted in an oil accumulation of about 5% in the stem. We think the theoretical maximum is about 20%, but that's quite an achievement because the wild type stem contains about 0.1% oil. And sugarcane can produce in the United States 60 tons of dry matter per hectare. So this will be a large amount of oil. And of course we know how to grow sugarcane, we know how to harvest it, we know how to process it. So the technologies are there if we can build the plant to do that. This was from a techno-economic analysis, which basically this shows the marginal land which could be available for such an oil crop. This is discounting Hawaii and Puerto Rico, which will be ideal for a crop like this as well. That will be enough to produce about 2/3 of the jet fuel use of the whole of the United States. And this is just point out, of course, Brazil has much more potential in this area and could really be a powerhouse for providing renewable fuels of this type. So with that I've finished. If you want to know more about the projects I've told you about, we have a website, right.illinois.edu, and of course, as I say, this wouldn't have been possible without many collaborators, past and present postdocs and graduate students, and this is an incomplete list of all the people that have contributed to that. So thank you. I'm happy to answer questions, but I notice there's some biofuel in the corner.
Who'd like to ask a question? Yes. Please, can you speak into the microphone even though it's not on?
Thanks. Mike Coe from Planet Hydrogen from Manchester. From the point of view of climate change, which of course we're all concerned with here, if you're making jet fuel even very efficiently, you're taking carbon from the atmosphere, processing it through plants, and in burning it, returning it to the atmosphere. What is the environmental advantage of that?
Well, it saves you. It saves you taking it out of the ground and putting it into the atmosphere, which is what we're doing today. Do you have an alternative?
Yes.
No.
Basically, if we don't carry on aviation, we have to use hydrogen. Okay, but all the same, you're simply replacing fossil fuels. If the danger to climate is not emissions, but concentration, you're maintaining concentration by using biofuel, not reducing concentration.
Yeah, I'm not claiming to do that, but how quickly are you going to convert the whole fleet of aircraft, of military aircraft to hydrogen? You're going to do that next week? I mean, where is your hydrogen plane? You know, are you going to have these planes by 2050? Yeah, but you know, we were discussing this before this. Bill and I have heard these stories for the last 40 years. Are we going to go on talking about the perfect getting in the way of the good forever? So,
Any more questions?
I've got a question about the QE quenching. It's a bit technical, but
yes.
there has been discussion about using mutants, the PSPS mutants, unable to quench, to use them in greenhouse conditions, so that if you have optimal controlled growth conditions, in principle they should be able to grow better. If you don't need to under non-fluctuating conditions. So I was just wondering if there's any evidence for that.
I'd say, yeah. Certainly, when we've grown these plants in fully controlled environments, there's no difference between the engineered plant and the wild type plant. So. Oh, that's the work at Nottingham. So, yeah. I mean, certainly, you know, of course they could quench, but we down-regulated PSPS hugely with RNAi. We didn't eliminate it. And, and under constant conditions, those plants are identical, actually.
Other questions?
So I understood that in your first example, plants didn't have time enough to adapt to higher CO2 concentrations. But in the second example, as far as I understood you, the case was that plants weren't regulating quickly enough for the changes in light intensity that comes from shading from higher leaves.
Yes.
But wouldn't that have been the case over millions of years, and shouldn't then the plants have time to adapt to leaves above other leaves?
Yeah, that's a great question. But I mean, there are two aspects to that. One is, of course, we're supposing that productivity is important for evolution. And, you know, there are lots of examples. For example, some of you may be familiar with Arabidopsis, this tiny genetic model. It's a very successful plant in the wild, but very unproductive, very low photosynthetic rates. The other one is that we actually think if you look at crop ancestors, like the ancestor of soybean, you know, grows typically in quite dry, nutrient-poor environments. Most of its leaves are exposed to full sunlight, so there's very little shading. If you look at the ancestors of wheat, for example, growing, you know, as they do in Syria, the Golan Heights, again, they're growing on these dry areas, quite nutrient poor, very sparse canopies. So the leaves are really not shaded. We think the shading, you know, the idea of crops having 6 layers of leaves is a relatively recent construct, you know, due to the way we plant them at high density under fertile conditions. So we think actually the crop ancestors didn't face the problem that our crops today are facing. We don't know that's the answer, but that's our speculation.
Just following up, then if outside of crops then, are there no plants or other photosynthetic species that would have been environments where this kind of advantage, like having a larger faster turnaround of, non-photochemical quenching or increased PSBS would have been there. Nobody ever looked for that or found it in the right environments, that is to say, where these kind of conditions occur?
No, I think that's a really good question, and it's actually motivated Chris and I to look at this because, because of course we've got part of that recovery, but there's still more to be had there that we can't get from these 3 genes. And so one of the things we're now trying to do is looking at plants. Well, obviously forest environments is one place where we expect this to be happening, is then looking at those to see, can we see faster recovery there, and what makes those plants different, genetically. So
Any more questions?
Thank you. You started talking about, in your presentation, talking about the projections for future yield. And one of the things which is apparent in the work the IPCC is doing, looking at sort of future land use scenarios, is there are some very heroic assumptions built into some of the kind of the models about future yield increases, future dietary change. I suppose my question is, at what point does the sort of work that you're doing, what point would you sort of been confident about feeding that into the larger global scale models and thinking that actually this could be the point in time where you could change those projections?
I think I guess I'd say it's too early at this stage. I think that what we're now looking for, what we really want to see now is, if we get this into cowpea, into rice, does it really result in a yield increase? And where does it? So we would then want to grow these in some different environments. So I'm afraid I'd say it's probably 5-10 years down the road before we even have data that could then say, okay, yeah, you should put this into the models because we can really do this now. Sorry.
Any more questions? Is it another hydrogen question?
No. Professor Long, I remembered a staggering dissertation you gave practically at the Royal Society 10 years ago on the dangers of ozone to all crop productivity. You can't have forgotten that. In improved productivity, for example, at Illinois over the next 20 or 30 years, ozone must come into it. Have you reflected on the relationship between these two, especially in rising ozone levels?
Well, I guess, in fact, following on from that, we published a paper in PNAS where we actually, we've now had ozone monitoring in the United States long enough that we were able to statistically back out what the crop loss is over the United States. And today it's around about 8% of the maize crop is lost due to ozone, about 4% of the soybean crop. What we were also able to show is that as the new emissions regulations came in, it really did make a difference on the historical data. So the losses are diminishing, but I'd say globally I'd expect the losses to be increasing because elsewhere in the world there's a lot more fossil fuel combustion going on. Fossil fuel combustion is certainly, if we're using the high temperature that results in NOx production, then that is impacting crops. And in fact, my colleague, who was an author to the paper you mentioned at the Royal Society, Lisa Ainsworth, has been looking very intensively now at maize genetics, and has shown there's quite a lot of genetic variation there in ozone tolerance, and that is now being used in breeding. But I'm not saying ozone isn't an issue we've got to pay attention to. And certainly I'm hoping our use of combustion is going to go down with time. But at the same time, I think we've got to accept the fact that if we're looking for the perfect solution all of the time, we're never going to do anything. And I feel we haven't done a whole lot in 40 years. That's how long we've known about this problem. And we're going to tell the next generation they should look for the perfect solution before they do anything. So,
Last question, anybody? If not, I'll just give a huge thank you to Steve for incredibly stimulating and interesting, and I've learned a huge amount. Well, that's not surprising, but huge amount. Thank you very much for giving up your time and coming.
well, thank you.